The overall goal of this procedure is to prepare a sample for cryo-TEM by extracting a focused ion beam lamella from a cryogenically frozen bulk sample. This is accomplished by first plunge freezing the sample in liquid nitrogen and mounting it inside the cryo-focused ion beam SEM. The second step is to cut out a thin lamella with the ion beam and transfer it onto a TEM grid by means of a cooled nanomanipulator. Next, the TEM grid is transferred from the SEM holder to the TEM holder in a transfer station. The final step is transfer of the TEM holder to the cryo-TEM for further analysis. Ultimately, the cryo-TEM is used to show high-resolution images, tomograms, or X-ray maps of the extracted lamella.
This technique will enable us to analyze soft matter with ultimate resolution in the transmission electron microscope by pre-selecting the place of analysis by other microscopies such as light microscopy or scanning electron microscopy.
The main advantage of this technique over existing methods like cryomicrotomy is that preparation artifacts such as compression and deformation of the sample are avoided.
First, mount two TEM grids for focused ion beam samples on the SEM transfer holder. Secure the grids by tightening the corresponding screws with a screwdriver and close the shutters. Mount a sample stub appropriate for the specimen and add a portion of the specimen. For example, use a pipette to deposit droplets on a flat stub. Then, mount the SEM transfer holder onto the vacuum transfer device or VTD.